Hello everyone. Welcome to this episode in our pharmacology series. Today we begin our journey into pharmacodynamics, a core concept in understanding how drugs interact with the human body. By the end of this session, you'll exactly know what pharmacodynamics means, how it's different from pharmacokinetics, and the basic ways in which drug produces their effects. Now, what is pharmacodynamics? Pharmacodynamics literally means the power of a drug. It refers to the effects that the drug has on the body and how these effects are brought about. It's not just about what a drug does, but how it does it, whether by binding to a receptor, modifying an enzyme, or altering a cellular process. In pharmacodynamics, we differentiate between drug action and drug effect. Drug action refers to the initial interaction of a drug at its target, like binding to a receptor or inhibiting an enzyme. But what we actually see in a patient, like reduction in blood pressure or pain relief, is a drug effect. This results from a series of downstream events following the initial interaction. Now, let us see mechanism of drug action. So, how do drugs produce their effects? Most commonly, drugs work by binding to a specific receptor. Some drugs, like aspirin, act by inhibiting enzymes, while others modify ion channels or simply act through chemical interactions, like antacids neutralizing stomach acid. Now, let's understand different types of drug actions. Drug actions can be grouped into five main types. First, stimulation, like how adrenaline increases heart rate. Second is inhibition, where drugs block natural processes like neostigmine preventing breakdown of acetylcholine. Third is irritation, which is mostly local and often undesirable. Then is replacement, like insulin therapy in diabetes. And cytotoxic action, which directly destroys abnormal cells, common in cancer treatment. Now we'll explore one of the most fundamental concepts in drug action: how drug interacts with the receptors. What are receptors? Receptors are specialized biological structures, mostly proteins, that recognize and bind to specific molecules like hormones, neurotransmitters, or drugs. They can be found on cell surface, inside the cytoplasm, or even in the nucleus. When a drug binds to its receptors, it sets off a chain of events that leads to physiological response. Let's know the type of receptors. Receptors can be classified by structure and location. The four major types of receptors are ligand-gated ion channels, G protein coupled receptors, enzyme-linked receptors, and intracellular nuclear receptors. Now, how drugs bind to receptors? Drug binding to receptor is usually selective, reversible, and follows the lock and key model, where the drug fits the receptor precisely to initiate a biological response. Two key properties define the interaction. First is affinity, which is how tightly the drug binds. And the second is intrinsic activity, which reflects the drug's ability to activate the receptor once it is bound. Drug can act in various ways depending on their interaction with the receptors. An agonist fully activates the receptor. A partial agonist binds but produces a weaker response. An antagonist blocks the receptors without activating it. An inverse agonist reduces the receptor's basal activity, doing the opposite of an agonist. In some systems, you don't need to activate all the receptors to get the maximum response. These extra or spare receptors allow the body to amplify signals, meaning a small amount of drug can cause a large effect. With prolonged stimulations, receptors can become less responsive. This is called desensitization. With chronic exposure, receptors may be downregulated, meaning their numbers decrease, reducing drug efficacy. This is seen in long-term use of beta agonists in asthma, leading to decreased responsiveness. Understanding drug receptor interaction helps doctors to select the right drug, predict its response, and tailor the dose. It also explains phenomena like tolerance, resistance, or drug failure in some patients. To summarize, drugs bind selectively to receptors, which then activate downstream pathways to produce pharmacological effect. Understanding these interactions is key to mastering pharmacodynamics. So, today we have seen what are the basics of pharmacodynamics. In our next lecture, we will talk about dose response relationship, potency, efficacy, and therapeutic index. So, see you all in the next lecture. Thank you.